Hello, everyone, and welcome to today's webinar, Maximizing Tumor Microenvironment Analysis for TILs from Limited Patient Samples. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All of your questions will be answered via email following the broadcast today. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. César Evaristo, Group Leader R&D, Tumor Immunology at Miltenyi Biotec. Dr. Evaristo, you may now begin your presentation.
Welcome you all to this webinar entitled Every Cell Matters, How to Maximize Tumor Microenvironment Analysis from Limited Patient Samples. I will start by giving a short introduction, followed by identifying some of the challenges faced when conducting cancer research and how we developed workflows to overcome those challenges. I will then zoom in on some of those workflows and detail some of the tools we use for sample preparation, magnetic enrichment, and analysis of the populations of interest. The immune system has the capacity to eliminate tumors. Mutated antigens can be released from tumor cells and induce an immune response in which DCs will activate tumor-specific T cells. These T cells then migrate through the blood vessels into the tumor sites where they recognize and kill the tumor cells. For the most part, this is an effective process. However, in every single cancer patient, at least one of these steps have failed. Immunotherapy has proven clinical efficacy and tremendous potential in multiple cancers, but even patients who experience clinical benefits from these new agents often achieve only partial responses, such that additional work is necessary to improve immunotherapeutic strategies. As this figure illustrates, solid tumors are complex tissues. Not only are they comprised of proliferating tumor cells, but are supported by a plethora of stromal cells that participate in functions such as tissue remodeling and neoangiogenesis, including many different immune cells. All of these cell populations are in constant communication and influence each other's activity, function, and fate. It's becoming increasingly clear that a comprehensive analysis of the tumor microenvironment is key to understand the dynamics of the anti-tumor immune response. In our case, we're interested in phenotyping tumor cells, fibroblasts, endothelial cells, T cells, B cells, and myeloid cells, and sequencing tumor cell mutations, identifying new antigens, and tumor-specific T and B cell clones. However, to do this, we are dependent on the availability of tumor material. This brings us to the challenges routinely faced when planning and performing experiments in cancer research. Most of the tumor samples from patients we get weigh between 0.4 and 1 gram. After dissociation, we get between 4 and 20 million live cells. It turns out that the frequency of TILs, or tumor-infiltrating leukocytes, in the tumor can be very reduced, as illustrated by this graph on the left. I calculated in this table on the right the absolute number of T, B, and myeloid cells that we would obtain from a tumor with 20 million live cells containing the average frequency of that we have observed among our samples. We'd get 1 million T cells, 200,000 B cells, and 1.8 million myeloid cells. However, if we have to divide our dissociated tissue in multiple samples to individually analyze the different cell populations, we would waste the majority of our cells. To prevent that, we developed tools and workflows that allow extracting the maximum amount of information from limited material, while minimizing the experimental time and workload. I will start by briefly summarizing examples of our strategy and then zoom in on specific portions of it. It all starts with the resection or biopsy of the tumor sample. The tissue is stored and transported in tissue storage solution. Upon arrival, a small tissue section is collected for future immunochemistry analysis. The remainder is dissociated, and we take a small sample to identify and quantify the immune cell populations. We can then isolate T cells using Realis CD4-CD8 TIL microbead kit. These new reagents can be released from the cell surface after positive selection of the cells of interest. The isolated T cells can then be used for phenotypical analysis, undergo further magnetic removal of dead cell in order to obtain cells with high viability, which is required for downstream applications such as single-cell immune profiling and RNA sequencing, or even be used, for example, for further magnetic subset enrichment, followed by in vitro expansion and further analysis. The negative fraction from the T cell isolation can also be used. The Realis CD45 TIL microbead kit can be used to isolate the rest of the tumor-infiltrating leukocytes. After magnetic release, these cells can be further used to isolate, for example, myeloid cells or B cells, which can then be used for downstream applications such as phenotype, culture, or sequencing. The negative fraction from the CD45 isolation can also be used for tumor cell enrichment. This is performed using a negative selection kit, and the tumor cells can then be used for multiple downstream applications, such as next-generation sequencing and new epitope prediction, phenotype, culture, or establishment of patient-derived xenograft models. Finally, the positive fraction from the tumor cell enrichment step, other cells of interest can be recovered, such as cancer-associated fibroblasts, which can be phenotyped. I will focus the rest of this webinar on a subset of these workflows. Additional Miltenyi webinars detail the performance and downstream applications of the other workflows. I will do that by showcasing some of the analysis we performed on one particular sample from an ovarian cancer patient. Most of the data going forward was generated from a single tumor sample. Additional supporting data used to characterize the performance of particular tools will be clearly identified. In this particular experiment, we were contacted in the middle of the afternoon with information that an ovarian carcinoma sample had been resected from a patient. The tumor sample was stored in MAC's tissue storage solution and shipped to us overnight. This solution has been developed to allow optimized storage of fresh organ and tissue samples. It has been tested and validated on a variety of human and mouse tissues, including tumors, lung, heart, brain, spleen, skin, kidney, and skeletal muscle. The graph on the left shows the frequency of cells per gram of tissue recovered after storage in different solutions for 48 hours at 4 degrees or room temperature, as compared with the fresh tissue on the column on the right. And this graph shows the viability of the cells in the same conditions. As you can see, tissues stored in MAC's tissue storage solution at 4 degrees maintain original cellular yield and viability even after 48 hours, with no induction of apoptosis or increase in necrosis, and no cell activation or influence on marker expression. The next morning, we used our automated dissociator and our optimized tumor dissociation kit to generate a single cell suspension. Using these tools, automated dissociation of multiple samples can be done in parallel. It combines both mechanical and enzymatic treatment for efficient dissociation. This means the disruption of extracellular matrix and cell adhesion components without destroying the integrity of the cell membrane, while simultaneously preserving the cell surface epitopes. The interest of optimized enzymatic dissociation can be showcased in this example. Just doing a mechanical dissociation will result in the loss of cell populations which are closely attached to the tumor cells, such as tumor-reactive T cells. Using the tumor dissociation kit will enable the identification of these rare cell populations. Furthermore, we do in-house control of the quality of the enzymes used, and the tumor dissociation kit was optimized for epitope preservation. We tested over 200 epitopes, the vast majority of which was not affected by our enzyme composition. I'm not expecting you to be able to read this, but this list of tested epitopes can be accessed on our website. After dissociation, we took a small sample to identify and quantify the immune cell populations using our 8-color immunophenotyping kit. Here, I am showing the plots with the gating strategy. After excluding dead cells in doublets, we gate on CD45 positive leukocytes. From those, we use a morphology gate to exclude small debris in large tumor cells. We can then identify CD14 positive macrophages, and from the CD14 low population, we plot CD19 versus CD3 to identify B cells. From the non-B cell gate, we plot CD16, CD56 in the same channel against side scatter to identify neutrophils and eosinophils. From the side scatter low gate, we plot CD16 and CD56 against CD3 to identify NK cells, T cells, and CD3 positive, CD56 positive cells. Finally, from the T cell gate, we plot CD4 versus CD8 to identify our major T cell populations. We can then quantify the frequency of the major leukocyte populations in the tumor, and it becomes quite obvious that the frequencies of each of the individual populations is quite low. Because these cells are so often found in low frequencies, we developed new reagents for the magnetic isolation of T cells specifically from the tumor based on the new Realis technology. Briefly, the Realis technology relies on recombinantly engineered low-affinity antibody fragments that are multimerized in the Realis complex. Cells are labeled with these biotinylated complexes in anti-biotin microbeads. The target cells are then retained on a column placed in a magnet, while non-target cells flow through. The target cells are then eluted and the microbeads are removed, resulting in bead-free cells that can be used for second magnetic labeling in isolation. For certain applications, the Realis complex can also be removed, resulting in label-free cells. Using these new reagents, we were consistently able to quickly isolate tumor-infiltrating leukocytes with high purities and directly use them for analysis. Or because of the additional flexibility provided by the Realis system, we have a world of possibilities for isolation of specific populations. We can purify Tregs from tumors, combining Realis CD4 TIL with CD25 microbeads. We can generate high viability TILs for RNA-seq using dead cell removal kit after T cell isolation. We can isolate tumor-resident memory T cells, isolate tumor-specific versus bystander T cells. We can isolate untouched gamma delta T cells, and so on. We isolated T cells from our dissociated ovarian carcinoma sample using Realis CD4-CD8 TIL microbead kit. In addition to this, we also have available Realis CD4, CD8, and CD3 microbead kits for tumor T cells. And we will soon also have the Realis CD45 TIL microbead kit available. Using these brand new reagents, we were able to quickly isolate TILs with high purities. On the top row, you can see the frequency of CD3 positive cells among live single lymphocyte-sized cells in the bulk sample as compared to after isolation on the bottom row. We were able to achieve over 92% purity of tumor-infiltrating T cells from our ovarian carcinoma sample. After magnetic enrichment of our cells of interest, we are now ready for analysis. This is done using high-affinity antibodies and analyzed on the MACsquant, a fully automated flow cytometer. High-affinity antibodies are genetically engineered to enhance reproducibility and eliminate bias. All antibodies have one universal IgG1 isotype, which reduces the complexity of the experimental planning, and a mutated Fc region that eliminates Fc receptor binding mediated background. Using magnetically isolated T cells from the ovarian tumor sample greatly facilitated flow cytometry analysis. Having highly enriched cells of interest reduces the number of total events we need to acquire for meaningful analysis of rare subpopulations, resulting in shorter acquisition time, reduced background, and cleaner populations. In this example, we interrogated the expression of PD1, CD39, TIM3, and 41BB among CD4 T cells on the top row and among CD8 T cells on the bottom row. In addition, we were interested in performing RNA-seq and immune profiling T and B cells infiltrating this tumor sample. We cryopreserved an aliquot of the dissociated tumor, and at a later time point, we thawed our sample and removed the dead cells using the dead cell removal kit. We then used the Realis CD4-CD8 TIL microbeads to isolate T cells for single-cell TCR sequencing and RNA-seq. From the negative fraction, we isolated B cells and plasma cells for single-cell BCR sequencing. This way, we could use the majority of T cells for the T cell analysis and the majority of B cells for B cell analysis. We used the 10X platform, which can generate up to 10,000 single-cell gems per sample. So a direct effect of magnetically isolating the cells of interest is that we greatly increase the frequency of single-cell TCRs and BCRs on our sample, as you can see in this table. In these graphs, I am displaying in the x-axis the 50 or 25 more represented clonotypes in T or B cells respectively, and in the y-axis the number of cells that express that particular clonotype in the isolated sample. As you can see, we were able to identify several clonotypes that were represented multiple times in the isolated samples. In contrast, several of those clonotypes would have gone unnoticed when we analyzed the bulk samples. For example, a clonal type that was present in 28 T cells in the isolated sample was only present in 3 cells in the bulk sample. On the B cells, we were able to identify one clonotype represented 120 cells from magnetically isolated B cells, but was only found in 4 cells in the bulk sample. These results highlight the degree to which magnetic isolation can improve the resolution of single-cell RNA-seq and immune profiling. With this workflow, we were able to identify and quantify tumor-infiltrating immune populations, isolate multiple cell populations for downstream analysis, characterize phenotypically the isolated populations, and increase the resolution of single-cell RNA-seq and immune profiling of cells of interest. However, in order to completely characterize the tumor microenvironment, it may be useful to access the spatial distribution of different cell populations and their phenotypes. In order to do that, before tumor dissociation, we collected a small tissue section and used our MACsima platform, which allows the staining of hundreds of markers in a single sample. It uses the principle of an iterative staining process conducted by the MACsima imaging platform in a completely automated manner. Briefly, this platform stains the sample with a first specificity and acquires a fluorescence microscope image. The staining is then erased. The sample is stained with a second specificity and a second image is collected. This cycle can be repeated hundreds of times in a fully automated way. As an example, I am showing a section from our initial ovarian cancer sample with a region rich in tumor islets in the top right corner, characterized by expression of EpCAM and T-SPAN8, in a stromal region without tumor cells. In the following slides, we will keep EpCAM staining in red to identify tumor cells, and the indicated marker will be displayed in white. Looking at CD45 expression, we can appreciate that immune cells tend to accumulate outside of the tumor area. We can locate the macrophages using CD14 expression, for example. CD38 identifies B cells. Plasma cells can be identified by CD138 expression. CD3 expression identifies T cells, of which the majority are CD4 cells, consistent with our flow cytometry data. But also we can find our CD8 T cells, and we can further characterize phenotypically the T cells. In this case, with the expression of the co-stimulatory molecule CD27 or CD28 or 1BB, ICOS, but also markers that correlate with tissue-resident memory T cells like CD69 and CD103. Expression of chemokine receptors can also be assessed, like CXCR3. We can identify cells that express PD-L1 for negatively regulating T cell activity, and the expression of immune checkpoints like CTLA4, PD-1, and TIM3. We can even identify which cells were actively proliferating using Ki67 staining. In this case, unfortunately, this indicates that tumor cells are the ones proliferating in this sample. In summary, I showed you here today new workflows that allow the complete characterization of the tumor microenvironment. These start with tissue preservation during shipping with tissue stored solution, automated tissue dissociation with optimized enzymatic mix, and identification and quantification of tumor-infiltrating immune populations. By implementing magnetic enrichment steps, we are able to analyze multiple distinct populations while minimizing the loss of limited material and enhance the phenotypic characterization of the isolated cell populations. Using the new Realis technology, we can perform consecutive enrichment of dedicated TIL subpopulations and have complete flexibility for studying several cell populations from just one sample. In addition, magnetic enrichment results in increased resolution of single-cell RNA-seq and immune profiling of cells of interest. Finally, with MACsima Imaging Platform, we can study the spatial distribution of different cell populations and their phenotypes within the tumor microenvironment. Using these workflows, it becomes feasible to fully embrace the complexity of the tumor microenvironment. I would like to acknowledge my team and all my colleagues at Miltenyi Biotec that made it possible to put together these workflows, as well as our collaborators at the University of Göttingen, Bonn, and Cologne for providing material and testing our workflows off-site.
Thank you, Dr. Evaristo, for your informative presentation and for your time today. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. All questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.